Though our brain does some impressive stuff prenatally, it also does some impressive stuff after we are born. In this module, we'll learn about the human postnatal cerebral development. The majority of our studies and research of the development of the brain comes from the study of non-human species. However, one way in which the development of human brain seems to be unique is that the brain of humans develops significantly slower than those of other species. This does not achieve full maturity until late adolescence, whereas if you look at a horse that would have been born just a few minutes or hours after, it is in various ways autonomous and it reaches independence at a much younger age. If you've interacted with an infant and a toddler, you would see how they are able to reach milestones of being able to sit upright and eventually to babble and eventually to walk and eventually defy gravity within the first 2 years of their lives. We find that this begins to slow down as we get older. As the volume of the brain continues to increase between birth and adulthood, please note that the substantial increase in size does not result from the development of additional neurons. As you learned in previous modules, actually neurons are dying, and that's what we call synaptic pruning. When synapses are made, we call this synaptogenesis, and this would be brought about by being stimulated by an individual. So we find that a lot more connections between neurons in specific regions of the brain can be an indicator of a child's analytic ability. You find that a child would be easily amused and entertained by the same joke, a peekaboo, whereas an older child would not be amused by that. And that would be a result of a child maturing. Now, the maturation process, the maturity would be brought about by synaptogenesis. The myelination of axons, which, as you already know, increases the speed of axonal conduction, also occurs in sensory areas and motor areas, as you may have learned and remember from general psych. According to Piaget, we learn the world through our sensory motor stage first. We do not understand language, nor do we speak, so it is through learning, sometimes by burning ourselves, that we learn that things are not supposed to be done. So by enhancing our senses and our movement, we are able to adjust and adapt to the world. Please note that this myelination continues into adolescence, specifically at the prefrontal cortex, and that's where you and I make a lot of the decisions, the higher executive functions as to what to do. Think of a 5-year-old who may just speak their mind, and if someone's breath smells, that 5-year-old may say it directly to their face. As we start getting older, we start realizing that even though there may be someone with not so pleasant breath, rather than offending them, we may kind of just breathe through our mouth and not be offensive. So this would be a result of more myelination at the prefrontal cortex. And the reason why is because at that point we would have some type of filter in there that would say, like, it's not nice, it may be offensive, so don't say anything. Whereas a 5-year-old that does not have this myelination at the prefrontal cortex may not be able to refrain themselves. We also find that increased branching of dendrites will progress from the deeper parts of the brain to the more superficial layers. And we're talking about the central core, the reptilian brain, the old brain, into the new brain, neocortex. This type of expansion would increase about voluntary behavior. Initially, it seems that you and I engage in a lot of repetitive or imitating behavior. If you've ever been in a nursery where there are a lot of babies, you find that sometimes there's a domino effect. If one child begins to cry, it may not be long before the second child begins to cry, and then the third and the fourth. And it may not be that they're uncomfortable, it's just that they're responding in a way that would be a little bit more imitating. Whereas voluntary behavior would be something that would be a little bit more sophisticated, where they may be able to discriminate something from something else. So they begin to imitate person A and not person B. This all an example of how our brain continues to develop. There are many opportunities for experience to influence the development of the brain. The effects of experience can be many and in a variety of ways, but they are considered to be of two general types, either permissive or instructive. Permissive experiences are those that permit the information in genetic programs of brain development to be expressed and maintained. For example, think back to chapter 2 where there were these male birds that had to learn a song every spring. They were born with the genetic program in their brain development to be able to sing, and it was consistent based on their ability to listen, but also it was renewed on a yearly basis. So because their brain is capable of doing this throughout their entire life, the genetic programs of the brain allows them to express and maintain that on a regular basis. Instructive experiences, instead, are those that contribute to the information in genetic programs and influence the course of development. In this case, we would not see a genetic predisposition of the individual having that, but instead the individual would learn that. And so perhaps this could be habits, or it could be something environmental, or it could also be the cells that just simply adjusted to stimulation that the environment provided. A very important feature of the effects of experience when we develop is that the development is consistent or dependent on when it happened. That is to say that they are time dependent. The effect of a given experience on the development depends on when it occurs during development. You may have heard of critical periods and sensitive periods, which are windows of opportunity in which a particular experience can influence the development. We find that if it is essential, crucial, for an experience to occur within a particular interval to influence development, then the interval is called a critical period. And by interval we mean time. If an experience has a great effect on the development when it occurs during a particular interval, but can still have a weak effect outside the interval is then called a sensitive period. Please note that although the term critical period is commonly used, the vast majority of experiential effects of development have been shown to be sensitive periods. An example of critical period is language development. We do know that we have a window of opportunity to teach kids how to speak, and that is typically by exposing them to the sounds and the phonemes. This occurs during the first years of their life. Ideally, in the first 2 years, they make amazing progress in terms of acquiring vocabulary. After that period, if we miss it, we can still teach them language, but it may be a little bit more difficult for them to learn that. And so think of individuals that you may know that have been in the country for 30 or 40 years, but depending on when they arrived, if they arrived at a time where it was essential or they were at their prime time to understand language and learn phonemes, then they were exposed to English at a critical period, and they probably speak it in a fashion that's consistent with most Americans. However, if they got here after that critical period, they could still learn English, but they would not be in a sensitive period, which could complicate their understanding and production of the language. A lot of our research on the effects of experience on the development of the brain has focused on sensory and motor systems. This has been done so mostly on rats, and this has been, for the most part, easier when we deprive individuals of light. Rats who were reared from birth in the dark were found to have fewer synapses and dendritic spines in the primary visual cortex. This is consistent with our understanding of the occipital lobe and the visual pathway, as we know that light is essential for individuals to develop their ability to see. In fact, we know that the retinal cells continue to grow and proliferate at a rate that's still quite rapid even after we're born. And sometimes there are individuals that believe that infants tend to sleep sometimes with their eyes halfway open, and the reason why is because there is a constant need for light. Depriving them of light or early visual stimulation could bring about deficits in depth and pattern visions as adults. We also find that through other studies with enriched environment, is that rats that were raised in enriched and complex cages rather than by themselves were more likely to have thicker cerebral cortices, and that is basically the outer part, the new brain. They're also more likely to have greater dendrite development, and that is to say that they are able to make more synaptic connections. And we also see that they would have more synapses per neurons. Keep in mind that these are good to have, especially in our earlier development of stage. As we start getting older, then we need to specialize a little bit more for our behavior and cognition to be a little bit more smooth. Early studies of sensory restriction in rats have also been extended to human babies who were born with cataracts. Now, we don't deprive them of light, but if they're born with cataracts in both eyes, this would pretty much render them nearly blind. When these cataracts were removed between 1 and 9 months after birth, their vision was comparable to that of a newborn. Thereafter, some aspects of vision improved quickly, but visual deficits were still present 2 years later. So please keep in mind the importance of experience and catching them at the critical period, and if not the sensitive period at the most, if we want the changes to be brought about. Our understanding of the neglect or the lack of stimulation earlier in life has become a little bit more selective. Depriving one eye of input for a few days early in life, we believe, has lasting adverse effects on our vision if we deprive one eye instead of both eyes. Through a study that is illustrated in this picture here, we see what's known as the monocular deprivation on ocular dominance. And that is to say that only one eye was deprived of vision, whereas the other was not. We find that this will bring about changes in the way synapses connect, more specifically at layer 4 of the primary visual cortex. We find that altered exposure during sensitive period could lead to the reorganization of the synapses in here. You can appreciate the changes with the eye that was not deprived, and you can see how the axon is really kind of just projecting across the different layers. Whereas the eye that was deprived, you could see that it is not branching out and not developing any dendrites thereafter. Research shows that with many species, ocular dominance columns in layer 4 of the primary visual cortex, which is what is impacted most, are developed at birth. However, if we blindfold one eye for several days during the first few months of life, this reorganizes the system. And so we would find that there are the columns would be wider in terms of what they're receiving input from the deprived eye is decreased, whereas the columns that receive input from the non-deprived eye would be increased. There are some that did not believe that the neurons would change in structure because of how quick the effect were manifested. However, research has shown that there is in fact a massive decrease of axons and their branching. To the LGN, specifically for visual, for the eye that is deprived, and we see that it would not be the same for the non-deprived eye. So we now know that in fact there is a structural change when we deprive the organism from stimulus that it should be exposed to. Some of the most remarkable demonstrations of neuroplasticity come from studies with the effect of experience and the organization of the nervous system. We look specifically on sensory topographic maps. And so the first study that we look at here is the cross-modal rewiring experiments in which ferrets basically had axons, and the retinal ganglion cells where synapse, instead of the LGN, they were specifically through surgically synapse onto the MGN. You may remember that that is what controls the auditory system, and the LGN would control the visual system. What we found in the 1990s is that the experience of the visual input caused the auditory cortex of the ferrets to become organized in a retinotopic fashion. That is to say that, well, these individuals thought that these ferrets would not be able to see, but instead hear. It turns out that with the visual input, the auditory cortices, which were the temporal lobe, now was able to see, and that is neuroplasticity. We also see the second study of changing the input of the cortical topography in owls. So we find that raised barn owls with vision displacing prisms over their eyes, this basically led to a corresponding change in the auditory spatial map. For example, an owl that was raised wearing prisms that shifted its visual world 23 degrees to the right had an auditory map that was also shifted 23 degrees to the right, so that objects were heard to be where they were seen to be. And this, again, allows us to see how we shift our auditory map in the prism-exposed owls, and they were able to see and hear. The early music training influence, also seen in our textbook, has also demonstrated that early musical training would expand the area of auditory cortex that responds to complex musical tones. And that is seen with individuals that are exposed to early music training. This would bring about a change in the organization of the human cortex. Neuroplasticity was once thought to be restricted to the developmental period. Mature brains were considered to be set in their ways, the old thing of you can't teach an old dog new tricks, and capable of substantial reorganization. But now there is accumulation of evidence that has indicated that mature brains are continually changing and adapting. From a perspective in the biopsychological branch, evidence that experience can influence adult neurogenesis is very exciting. One line of research on adult neurogenesis began with a study of effects of adult rats and mice that were living in enriched environments. These were environments that would change with a lot of toys, with wheels and other rats. And it turned out that older adults living in enriched environments produced 60% more new hippocampal neurons than did adults that lived in non-enriched environments. And so hippocampal, we're talking about memory, we're talking about memory consolidation. However, before we start looking at the effect of how our environment can bring about some benefits to our hippocampi, we should be aware that the observed positive effects on neurogenesis in the adult rat depended largely, if not entirely, on the increases in exercise that typically occur in enriched environments. So it's not just living in a fancy place, but the type of lifestyle. This finding has brought about different intriguing implications, and that is that since the hippocampus is involved in some kind of memory, exercise may reduce or delay memory problems. And so there are individuals that have been studying this a little bit more aggressively since the early 2000s. Please note that the mature brain is constantly changing and adapting, so that's part of a living organism. And neurogenesis, which is the growth of new neurons, is seen even in adulthood, particularly at the olfactory bulb and the hippocampi of adult primates. Please note that hippocampi is plural for hippocampus, and we do have two of those. The number of new neurons added to the adult hippocampus per hippocampus is about 700 neurons per day. And we find that with an enriched environment and exercise, we could potentially promote more neurogenesis, as stated earlier. Exercising may reduce or delay memory problems. Tinnitus, which is a condition that is characterized by continuous ringing in the ears, is not unusual, sadly, for some of our veterans who have come back and been exposed to very loud environments, and they live constantly with this ringing. Everywhere they go, they're able to sense this. Interestingly, what we've found with the reorganization of the adult cortex is that This would produce a reorganization not only of the ear, but in the primary auditory cortex. In a study in 1998, Molnackel and his colleagues found that tinnitus, or you also hear it pronounced as tinnitus, produces a major reorganization in the primary auditory cortex. Basically, demonstrating how the effect of the constant ringing will bring about an effect specifically at the part in the brain that is responsive to that continuous sound. In another study that demonstrates something similar, we found that adult musicians who play instruments that are fingered by the left hand have an enlarged representation of the hand in the right somatosensory cortex, which would be the cortex that controls the left side. Remember, the right cortex would control the left side of our body and vice versa. So we find again that these adult musicians who played these instruments, like the violin, they would have a larger part of the cortex that would be active with the individual's left hand. Whereas if the individual was playing a guitar, perhaps with the right hand, doing the fret movement with the right hand, we would see a lot more activity on the left side of the somatosensory cortex. And the last of the studies that we see here is that skill training does lead to the reorganization of the motor cortex. This was demonstrated in 1994 by Rossini and his colleagues, where they showed that when they would anesthetize a particular finger of an individual, this would reduce their representation in the contralateral somatosensory cortex. And so if an individual has a lot of exposure to the fingers and a lot of flexibility and movement, then we would see a lot more activity in a larger part in the motor cortex for the hand in question. But if we were to anesthetize them or just keep them together with a tape and don't let them move, then we would see that part of the brain also shrinking and decreasing.